Welcome learners to the video learning organization of prokaryotic genome. DNA is the language of life. As we begin learning molecular biology, let us understand the difference between a gene and a genome. A gene is a segment of DNA that encodes for a protein, whereas a genome is the entire DNA or genetic material of the organism. Gene consists of a few hundred to thousands of DNA bases, whereas a genome consists of a very large amount of DNA bases, generally ranging in millions and billions. Gene encodes for proteins, genome encodes for proteins, what we call it as coding DNA or coding genes, as well as regulatory elements which are called as non-coding DNA. An organism can contain several genes. An organism contains one genome, that is the entire genetic material. The study of genes is called genetics. The study of the genome is called genomics. We all have learnt that humans have diploid genome, that is 2n, meaning two copies of each chromosome, donated one from each parent. Unlike us, a bacterial genome is generally composed of a single circular chromosome, and a bacterial cells reproduce by making clones of themselves, which we call it as binary fission. The mother cell copies its own DNA chromosome, then splits her cell in half, keeping one chromosome and giving one to the new daughter cell. Since there is only one copy of the chromosome, that is 1n, bacterial cells are considered haploid. Prokaryotes are cells which lack nucleus. Hence, their chromosome are not enclosed by a membrane but is instead located in nucleoid. The nucleoid is a cytoplasmic location of the bacterial genetic material. Though the nucleoid lacks a membrane, it still succeeds in packing the chromosome in a small area within the cytoplasm. Several proteins help in holding the chromosome in this nucleoid space, which is filled with genetic material and devoid of ribosomes. This nucleoid area generally takes up about one-third the interior volume of the bacteria. When we question about the necessity of packing the chromosome into a small area within the cytoplasm, we will have to explore the concept of supercoiling. As we tend to cut the circular chromosome and lay out in a straight line, it would measure about 1.5 millimeters. That might sound small, but the average bacterial cell is only 1 to 2 micrometers in length. That means the chromosome is over 500 times the length of the cell. If the chromosome was not packed tightly into the nucleoid, the entire cytoplasm could be taken up by the chromosome, leaving little room for other important processes like cell metabolism and protein biosynthesis. Hence, like eukaryotic DNA, prokaryotic DNA undergoes supercoiling, but it is not wound around histone clusters. In bacteria, the supercoiling uses the application of tension to twist the DNA molecule so it wraps around itself, creating loops. The folding of prokaryotic DNA is facilitated by nucleoid associated protein NAPs instead of histones. NAPs are proteins within the nucleoid that can bind to the DNA molecule, introducing bends and folds, and they are involved with processes such as DNA replication and transcription. Proteins are able to bind to the DNA and twist the chromosome into loops of about 10,000 base pairs. These loops are called looped domain structures, and as the loop piles up, they give the chromosome a flower shape instead of an open circular arrangement. Proteins involved in supercoiling. During the 1980s and 1990s, researchers discovered that multiple proteins act together to fold and condense prokaryotic DNA. In particular, one protein called HU, which is the most abundant protein in the nucleoid, works with an enzyme called topoisomerase 1 to bind DNA and introduce sharp bends in the chromosome, generating the tension necessary for negative supercoiling. Recent studies have also shown that other proteins, including integration host factor IHF, can bind to specific sequences within the genome and introduce additional bends. The folded DNA is then organized into a variety of conformations that are supercoiled and wound around tetramers of the HU protein, much like eukaryotic chromosomes are wrapped around histones. Once the prokaryotic genome has been condensed, DNA topoisomerase 1, DNA gyrase, and other proteins help maintain the supercoils. Then, talking about the genome size, an average bacterial chromosome is between 0.16 megabase and 10 megabase. 1 megabase is equal to 1 million base pairs, which are adenine, guanine, cytosine, and thymines. The length of the genome varies between different species of bacteria. Comparatively, free living bacteria have longer genomes and more genes than bacteria that are obligate symbionts and parasites. Because these obligate symbionts and parasites live inside a sheltered or limited host environment and cannot survive outside of the host, so over longer periods of time, these organisms start to lose genes that are not essential. Rather, some are able to utilize some of the host gene products, eliminating the need to maintain their own genome. So far, we have learnt that the DNA of prokaryotes is organized into a circular chromosome supercoiled in the nucleoid region of the cell cytoplasm. Now is the time to learn about operon concept. Operons are clusters of genes which encodes proteins that are needed for a specific function. So here in the slide, lac operon is given as an example. In bacteria and archaea, structural proteins with related functions such as the genes that encode the enzymes that catalyze the many steps in a single biochemical pathway are usually encoded together within the genome in a block called an operon and are transcribed together under the control of a single promoter. This forms a polycistronic transcript. French scientists Francis Jacob and Jacques Monod at the Pasteur Institute were the first to show the organization of bacterial genes into operons through their studies on the lac operon of E. coli. They found that in E. coli, all of the structural genes that encode enzymes needed to use lactose as an energy source lie next to each other in the lactose operon, that is lac operon, under the control of a single promoter, the lac promoter. For this work, they won the Nobel Prize in Physiology or Medicine in 1965. Thank you all and looking forward in the next learning session.